All right, chale ji, bismillah, let's begin. Okay, so as for the conclusion for B, pale purple solution. Burette solution हमने डाला है. Burette is supposed to be the test for proteins, right? And a protein test is giving us a pale purple solution. What can we conclude, bachcho? Can we conclude that proteins were present?
Yes, proteins are present.
Excellent, proteins present. ठीक है. और इसी तरह फाइनल में देखो सी में क्या है, ब्लू ब्लैक सोल्यूशन आ गया. द फाइनल कलर इज ब्लू ब्लैक. व्हाट इज दैट इंडिकेटिंग? दैट्स इंडिकेटिंग स्टार्च इज प्रेजेंट. ठीक है. सब टेस्ट का देखें, वी हैव पॉजिटिव रिजल्ट, वी हैव द फाइनल एंड कलर. सो दैट्स द कंक्लूजन दैट वी कैन मेक. लेट्स लुक एट द आंसर, व्हाट द आंसर हैज टू से. अच्छा जी, ये देखें जी जरा. अब since it's a 4-marker question, let's see what more detail did we need. A, reducing sugar present. ठीक है. अच्छा, अब there are some things that we need to understand. देखें, can you see these bold letters R, bold letter A? You need to understand that in the marking scheme, a bold letter R means reject. So if there's anything written and then you have a capital R in front of it in the mark scheme, that is indicating that that comment will be rejected. That word is not acceptable. और A का मतलब होता है award, that's permissible. इस चीज के लिए कैंडिडेट को अवॉर्ड कर दे. ठीक है. So look at the first conclusion, reducing sugar present. Okay. ये reducing sugar present है, उसने कहा है अवॉर्ड नेम reducing sugar. आप अगर नाम याद है, नहीं भी है तो कोई बात नहीं. खाली sugar लिख दिया है, guess what, that will not be acceptable. Why? Because this test is specifically for reducing sugar. Yes, you will study inshallah later in chemistry that there is another type of sugar which is non-reducing sugar. What the hell is non-reducing sugar? Guess what, you don't need to go into that detail. The point is, if you just write sugar, that's unacceptable. या फिर अगर आपने carbohydrate भी लिख दिया, वो भी unacceptable है. The Benedict solution test is for reducing sugar. Reducing sugar, and I repeat, reducing sugar. Okay. Now B, proteins present. ठीक है. Polypeptides भी present लिख दिया, तब भी ठीक है. Starch पे उसने कहा है starch जो है वो present है. Okay, the starch is present. Reject weak, reject some unqualified like reducing sugar, whatever the case is. So alhamdulillah, हमारा conclusion जो है वो बिल्कुल ठीक था, and we've completed table 3.1. Let's see what the examiner has to say. A part B1 के बारे में हम comments देख लेते हैं पहले. B1 वाला part क्या था, जहाँ पे हमने colors of indicators बताने थे, so colors of the reagents. Many candidates had obvious experience of the solutions used to test for food materials and were able to state their colors correctly. Okay, a few lost credit because they recorded colors that would be expected for positive results. Dekhen, when we're talking about iodine and its color is brown. That's the original color of iodine. If we're talking about Benedict solution, then we know that the original color of the solution is blue. If you're talking about Biuret solution, the original color is blue. If you're talking about ethanol, the original color is colorless. Okay. जब हम food test का final result बताते हैं, we tell the color change, which will start from blue, of course, which will start from the colors of the reagents themselves, but then the positive result will, ethanol के case में आपको पता है अगर fat हो तो milky white color indicates positive result. If the result is negative, then the color will be colorless. You understand? If the result is negative, then the color will be the color of the reagent. That's very important. तो यहाँ पे वो आपको बता ही रहा है कि a few lost credit because they recorded colors that would be expected for positive results. First part में तो उसने ये पूछा ही नहीं. उसने पूछा है कि reagents का अपना क्या color है. Okay, so let's see what he has to comment for the second part. Nearly all candidates identified the presence of starch correctly, although few wrote iodine was present. Most identified presence of reducing sugar. Some stated only sugar. मैंने आपको बता दिया ये गलत है, and therefore were not awarded credit. Most identified presence of proteins or polypeptides. A good number of candidates recognized that the green color of the Benedict's test or the pale purple color of the Benedict test indicated small quantities. I would rather use the word concentration of either reducing or proteins were present. ठीक है जी, क्या ख्याल है बच्चों, कुछ समझ में आती है कि पेपर में वो किस किस तरीके से पूछता है? Yes?
So, sir, like when we're pointing out like starch is present or reducing sugars are present, so do we write that in which quantity they are present, or do we just write
Okay,
the present?
first of all, don't use the word quantity. That's the wrong word. The scientific word is concentration. That's number one. Number two, question पे depend करेगा. Yeah, आपको, if you have to indicate the presence of or the absence, you just state the presence or the absence. And if you've been asked otherwise, then you will have to do accordingly. ठीक है? Does that answer your question?
Yes, sir. Thank you.
You're welcome. चलिए जी, आगे अगला question देखते हैं. This is May June 2013. Question paper 6-1. Question number 1. Some students carried out an investigation into the movement of food molecules from small intestine using a length of whisking tubing. If you remember, whisking tubing हमने पढ़ा था osmosis में. This is a semi-permeable sort of a bag. Okay. Students investigation कर रहे हैं. They are trying to simulate the movement of food molecules from the intestine using a whisking tube to represent part of the digestive system. Whisking tubing is made of a flexible, transparent material. चलें जी, flexible भी है, transparent भी है, और semi-permeable भी है, ये भी हमें पता है. So there are three properties that we know of the whisking tubing. Now let's look at the diagram first. Okay, when it comes to the diagrams in your exams, you have to understand कि diagram को देख के आपने उसको fully decode करना है. You have to literally, thoroughly study the diagram. So you have a large test tube of water. Then you have end of tubing securely tied. नीचे से गिरा बांधी हुई है, ऊपर से ट्यूब को क्लिप के साथ टाई किया हुआ है. And then whisking tubing filled with a solution containing reducing sugar, glucose, and starch. So you have glucose, which is a reducing sugar, and you have starch. So you've got two things. You've got two things inside the transparent semi-permeable bag. You've got the glucose and you've got the starch. Now let's look at the points one by one. One end of the length of tubing was securely tied to close it as shown in figure 1. ये देखिए यहाँ पे दिखा दिया. Okay. This tubing was filled with a solution containing both reducing sugar, glucose, and starch. We've already mentioned this for ourselves. Let's redo this. Number 3, the outside of the tubing was rinsed with water. Test tube का बाहर पानी से क्यों धोया बच्चों? क्या ख्याल है? What do you think? Let's be critical with this. Why do you think there has been a case where, you've been asked to rinse it with water? Why?
Sir, maybe to clean it so that we can see the color change.
I'm sorry, can you say that again?
Sir, to see the color change properly so that the test tube is clean.
To see the color change properly. ठीक हो गया. ठीक हो गया. Good. Okay. अच्छा जी. So basically to wash away all of that stuff. चलें जी. So the tubing was placed. Excuse me. Just give me a second, please. The tubing was placed into a large test tube filled with clean water and held closed and in position by a clip. चलिए जी, आगे चलते हैं. Samples of water from the large test tube were tested for the presence of reducing sugar and starch at different time intervals. Okay, so हम क्या कर, samples of water. Wait, so we need to put things in perspective. चीजें तो अंदर है whisking tube के. They're inside, but what he's saying in the question, samples of water. Water is on the outside. Okay, now this is strange and we need to understand this. Why is he taking samples from the water? Samples of water from the large test tube were tested for the presence of reducing sugar and starch at different time intervals. देखिए, let's talk about both starch and glucose. What's the Common thing for both starch and glucose, both of them happen to be carbohydrates. Starch is a large, complex, insoluble molecule. Remember, we've studied this in digestion, okay? Whereas glucose is a baby, small, simple, and soluble. Which one of them do you think will be able to pass through the whisking tubing? The glucose or the starch? What do you think?
Glucose.
The glucose. Why? Because it's soluble. So we can't expect the starch to come out to the water, can we? We can't, okay, because it's large and it's complex. हम नहीं expect कर सकते कि starch molecules are big molecules. They can't cross the semi-permeable membrane because they are small pores, okay? So describe how you would safely test for the presence of reducing sugar. चलें, ये तो simple सा question है. तो आप हम सबसे पहले क्या करेंगे? Again, I want you to just pause and think about it. Okay, what do you think we should be writing? Again, it's a descriptive glossary word and it's a 4-marker question. Describe how you could safely, आप देखें वो safely कह रहा है, इसका मतलब है safety का तो जरूर कोई ना कोई point होगा इसमें. Again, since it's the same, reducing sugar, तो आप देखें कि answer will almost revolve around the same idea. Okay, let's look at this mark scheme now. How many points has he given? 1, 2, 3, and 4. The first point is add Benedict solution. Benedict solution का word उसने देख लिया, you will get the mark. Then you heat it, okay? और heat के साथ आप safety वाला point mention करें, water bath, उसका separate mark है. और फिर देखें blue to green to yellow to red. Now there's one thing I want you to notice in the marking scheme, and that is in the third point, he has underlined the blue color. What that means is, and this is a very important principle from the exam perspective for food tests, that whenever you have to tell, you have to describe the color change and not just the final color. ठीक है? अगर मैं खाली red लिख दूं, खाली yellow लिख दूं, खाली green लिख दूं, that will be unacceptable. It has to be blue, then to green, either yellow, orange, whatever the case is, anything should be acceptable. ठीक है? So the reason why he has underlined blue is because it's important that you tell the color change, which is from blue to whatever the case is. बच्चों बात follow कर रहे हैं? Are you understanding? This is important point, okay? Let's see what the examiner has to say about this. Okay, this question was generally well answered. जैसे कि माशाल्लाह आप लोग करेंगे एग्जाम में. Most candidates identified Benedict's solution as the correct reagent and also suggested a suitable range of colors to indicate the presence of reducing sugar. Some candidates omitted to specify that the solution should be heated. बार बार देखें आप एग्जामिनर कमेंट्स में देख रहे हैं, जब भी reducing sugar का टेस्ट है and you don't talk about heating, you are losing marks because this is the only food test which involves heating, okay? But those who did often also refer to the use of water bath. Water bath happens to be one of the safety features. You can very well heat it under the Bunsen burner, but you don't do that. You do it in a water bath, thus scoring the extra credit for a relevant safety procedure, because that's what he said in the question. How would you carry out the test safely? A few candidates did not indicate that the Benedict solution is blue at the beginning and thus did not gain full credit. Again, the reason I'm making you do this is everybody knows and understands and knows the content of the food test, but when it comes to attempting the exam style questions, that's where it gets challenging. ठीक है? चलें जी आगे चलते हैं B part. Okay, again, describe how would you carry safely the test for the presence of starch. अब यहाँ पे description है और safely है. तो starch बेचारे में क्या चीज़ है? Iodine solution है. और किसी भी solution के लिए safety क्या है? Gloves पहन लें ताकि वो आपको irritant ना लगे. So let's see what the examiner has to say for this. Iodine solution turns blue. I'm sorry, color change तो मैंने बताया नहीं. Blue, black. Acha, there is one thing I forgot to mention. in the previous answer, safety में water bath के अलावा आपकी eye goggles भी हो सकती हैं, lab coat हो गया, head cap हो गया, eye goggles हो गई. Since you're dealing with the heating and solution, so things can pop up and come into your eyes, and that's dangerous. Okay. So iodine add करेंगे. The final color is brown to black because that's what the final color is. ठीक है. Blackish नहीं लिखना, वो reject कर रहा है. So remember that's not to be done. Let's see what the examiner has to say for this also. This part of the question was also quite well answered, with iodine solution being identified as a suitable reagent to test for the presence of starch. Fewer candidates noted that starting color brown compared with part A. Again, इस पे भी आपने कलर चेंज लिखना है. तो ओरिजिनल कलर आयोडीन सोल्यूशन का कलर क्या है? ब्राउन है. और जब वो प्रेजेंस शो कर देगा तो फिर हम क्या लिखेंगे? ब्लू ब्लैक. सो अगेन you have to write the entire color change and not just blue black. Okay, तो वो यहाँ पे यही बात कर रहा है. Fewer candidates noted starting color brown compared with part A. A few candidates incorrectly identified the color indicating a positive result being blue. There were almost khali blue ठीक नहीं है, it's blue black. That's the key word. There were almost no references to safety in this part, although credit could have been given if not already awarded in A1. उसने common एक marking point दिया है since both of these parts were common. Okay. चलिए जी आगे चलते हैं. Explain why the outside of the tubing was rinsed before it was put into the large test tube of water. I think somebody answered this for me already. It is to remove reducing sugar and starch, yeah, to ensure that there's no solution already inside the test tube, because glucose ने dissolve होके बाहर आना है और अगर हमने उसको पहले से साफ नहीं किया, then that would cause a problem. Okay, so that's why it's important. Let's see what the examiner has to say about this also. Credit-worthy answers refer to the removal of solution from the outside of the tube. Many candidates gave less specific answers that included the idea that the tube should be clean, such as to prevent contamination, remove unwanted substances. This type of answer was insufficient and thus gained no credit. Okay. चलिए जी अगला part interesting है. The results of the samples of water tested are shown. Sampling time 0 पे reducing sugar negative था, starch test भी negative था. 2 minutes के बाद, अच्छा by the way मैं diagram redraw कर लेता हूँ यहाँ पे. ये अंदर आपका वो है whisking tube है, ठीक है, और हमें पता है कि inside is starch plus glucose, और बाहर वाटर के अंदर सिर्फ क्या चीज आ सकती है बच्चों? Only glucose, right?
Glucose.
Good. अच्छा, अब वो आपसे कह रहा है कि 2 मिनट्स के बाद रिड्यूसिंग शुगर टेस्ट नेगेटिव था. वो टेस्ट के लिए सैंपल कहाँ से ले रहा है? टेस्ट के लिए ही इज टेकिंग सैंपल फ्रॉम द वाटर, द पोर्शन दैट आई एम कलरिंग एज वेट, द पोर्शन दैट आई एम कलरिंग एज येलो, सॉरी ऑरेंज. ये इस पोर्शन में से वो सैंपलिंग ले रहा है जो वाटर है, अंदर से नहीं ले रहा, ठीक है. सो आफ्टर 2 मिनट्स रिड्यूसिंग शुगर का टेस्ट भी नेगेटिव था और स्टार्च टेस्ट भी नेगेटिव था. व्हाट डज दैट इंडिकेट कि 2 मिनट्स तक has there been any glucose coming out of the whisking tubing? Yes or no? The answer is no. Why? Because the test is negative. अगर वो आ गया होता तो test positive होता. But after 20 minutes, guess what? The reducing sugar is positive, which indicates that glucose has reached the water, while the starch remains negative. Why does that remain negative? Well, because starch is too large to pass through. You understand? अब वो आपसे ये कह रहा है, suggest and explain what had happened during the 20 minutes to produce these results. How many marks? Wow, 5 marks. आपको समझ में आ गई. Now the only thing is how do you put this in words? So let's look at the mark scheme for this. देखिए हुआ क्या? Diffusion happened from higher concentration to lower concentration, yeah, down the concentration. What diffused? Glucose diffused. So why can't starch pass? Because it is too large to pass through. Okay? And of course, no energy is required. हाँ जी, diffusion का concept है कि नहीं है बच्चों? ठीक है? So let's see what the examiner has to say. A significant number of candidates simply restated the results, noting the sampling times and indicating whether or not sugar and starch were present at those times. This was then explained by suggesting that it takes time for starch to be broken down into sugar. These answers were not credit worthy. Why can't we assume that starch digest ho gayi hogi andar or glucose mein convert ho gayi hogi? Has he mentioned anywhere in the question that inside the whisking tube you have enzymes that convert starch to glucose? Is that mentioned anywhere in the question? It's not mentioned. So why do I have to? So why do I have to assume? You don't have to assume this. That's what he's saying in the examiner comments. Okay. Okay. Right. Yeah. This was then explained by suggesting that it takes time for starch to be broken down into sugar. These answers were not credit-worthy. Correct answers, जैसे कि आप लोगों के होंगे इंशाल्लाह, refer to the process of diffusion and the movement of substances from areas of high concentration to low concentration. Of course, talking in the context of glucose. Some candidates gain credit for recognizing the differences in the relative sizes of sugar and starch. See, this is the first thing that we brainstormed. Molecules. Credit could also be gained for recognizing that diffusion is a process that does not require energy, although few candidates stated this fact. चलें जी, आखिरी पार्ट भी देख लेते हैं इस क्वेश्चन का. Suggest giving reasons which part of your digestive system might be represented by the whisking tubing in the water in this investigation. जी बच्चों, क्या ख्याल है? देखिए, the answer is ileum. Why is the answer ileum? Because ileum में glucose की absorption होती है. And the water here can represent the blood basically. I know इसमें थोड़ा सा ज़रा आपके brain cells ज़्यादा fry होने थे, involved होने थे. Let's look at the comments of the examiner. Many candidates gained credit for correctly identifying the small intestine ileum. Some of the answers did not score were too vague or incorrect. Further credit was awarded for reasoning that this is part of the body where absorption of digested food molecules takes place, and candidates expressed this in numerous acceptable ways. Credit was also available for indicating that digested food enters the blood. Few candidates earned this credit.